Hey everybody, it's Mr. Umlor. Welcome to our first video on the unit on cell biology, the building blocks of life. As you can see, I've got my UC Santa Barbara t-shirt on today. That's where I received my degree in biology from, and it's where I took one of my favorite classes of all time, which was cell biology, just learning about all the little things that go into cells and how they function and what they do. I just really found that fascinating from the get-go. So let's get started. The first thing we want to talk about when it comes to cells is something called the cell theory, which is kind of a set of rules, if you will, for what cells are and where they come from. So the first part of the cell theory says that cells are the fundamental unit of life, which means that you can't go any smaller than a single cell and still be considered a living thing. Whether you're talking about things like bacteria, whether you're talking about eukaryotic cells, they are the fundamental units of life. All living things are also made of one or more cells. So again, bacteria are prokaryotic cells. They exist as single-celled organisms, whereas eukaryotic cells, they can exist as single-celled organisms or, like us, multicellular organisms. And then all cells come from pre-existing cells. It sounds kind of intuitive, but you have to remember these rules of the cell theory were developed many, many years ago when the first microscopes were being developed, and it was once thought that cells could spontaneously generate from non-living sources, which we now know is not the case. Another interesting thing about cells is the size of them. When you're comparing things like the cells of a human being to cells of, say, a blue whale, the largest animal that's ever lived, our cells are basically the same size. It's just that blue whales have a whole heck of a lot more of them than we do. Bacterial cells are going to be a lot smaller than eukaryotic cells, but they're all limited by one key thing, and that's the surface area to volume ratio. So just to give you an example, here's 3 sample cells. So they've got differing diameters. This one here has a diameter of 1 micrometer, this one is 2 micrometers, and this one is 3. The thing that you really want to pay attention to are these numbers down here at the bottom. Notice that as that diameter gets bigger and bigger and bigger, the surface area to volume ratio gets smaller and smaller and smaller. What that means is it makes it harder to get nutrients and other needed materials into the cell, and it makes it harder to get waste products out of the cell. Imagine a city that has 2 roads going in and out of it, and the population of that city doubles or triples, and the roads that are going in basically stay the same. The trash trucks are going to have a hard time getting the garbage out, and the food trucks are going to have a hard time getting food and supplies into that city. So it's the same sort of idea with cell size. And we'll be doing a lab here pretty soon to illustrate that a little bit better for you. Another key component of all cells is the plasma membrane. All cells have this membrane that separates them from their environment that they're found in. So it forms the outer surface of all cells. It acts as what we call a semi-permeable barrier. And what semi-permeable means is that it allows some things through and some things don't go through. So think of like a colander for making pasta. You pour the hot water with the pasta in it into the colander. The water goes through, the pasta stays behind. So that is a semi-permeable membrane. Same thing with our cell membranes. It allows some things to easily pass through and other things not quite so much. But we'll get to that in our next video. The cell membrane also allows for a maintenance of a more stable internal environment, so you can regulate things coming in and out to control the internal conditions of that cell. It also allows for communication between cells. So we'll get into cellular communication a little later on in this unit, but a lot of these structures that you see up here, these carbohydrates and some of these proteins and things like that, allow for communication between different cells. And then lastly, there are proteins in the membrane that allow the cells to bind and adhere to other cells. But again, we'll get to most of this in our next video. Next thing I want to talk about is the difference between prokaryotic cells and eukaryotic cells. This is a key thing in understanding cell biology. So when we're talking about prokaryotes, we're talking about archaea and bacteria, the more ancient and simple single-celled life forms on the planet. Prokaryotic cells are not compartmentalized, which we'll get to here in just a second, and they also lack a nucleus, which is what defines the difference between prokaryotes and eukaryotes. You'll notice on this side you have a nucleus, and over here all the DNA is just kind of floating around in the cytoplasm. So eukaryotic cells are going to include things like protists, plants, animals, fungi. Anything that's a multicellular organism is going to be eukaryotic. These cells are compartmentalized. So you'll see over here we've got this little guy here. We've got some other organelles that are organized into their own little compartments, whereas our prokaryotic cell doesn't have that. And we'll talk about the importance of that in a minute. And lastly, the defining difference between eukaryotes and prokaryotes is that presence of a nucleus or absence in prokaryotes. So very quickly, some characteristics that are common amongst all prokaryotic cells. The first is the plasma membrane. So if you look over here in this picture, kind of this first layer they're showing peeled away here, and then this one over here, that's the plasma membrane. Same sort of membrane you'll see in eukaryotic cells. Prokaryotes have what's called a nucleoid, which is this area here. It's kind of this white spot on our cell over here, which kind of actually makes a little bit of a face now, doesn't it? But the nucleoid is the DNA. It's the region in the prokaryotic cells where the DNA is found. It's not contained within a nucleus like eukaryotic cells. All cells have cytoplasm as well. And the cytoplasm, you may also see the term cytosol. Cytoplasm is everything inside of the plasma membrane. So all of the stuff floating around inside the cell. The cytosol is the watery, kind of liquidy stuff that all the other pieces are floating in. So these three things, along with ribosomes, are found in all prokaryotic cells. And we'll talk about what ribosomes do here in just a minute. Some other things that are found in prokaryotic cells, some of them, but not all of them. The first is the cell wall. So in this, again, if we look over here, this middle layer, kind of this brownish color, is the cell wall. It's different from a plant cell wall in the structure, but the function is basically the same to maintain the shape of the cell. And it's actually made up of molecules called peptidoglycans, which are protein carbohydrate combos, and it actually creates one giant molecule that surrounds the cell to protect it. Some prokaryotes also have a capsule, which is this outer layer out here. This is kind of a slimy kind of layer made out of mostly carbohydrates that protects the cell from things like your immune system cells. So anything that's infected by these bacteria, it's going to provide them some protection. It also helps to keep them from drying out. But again, not all prokaryotes have a capsule, and the ones that do can actually survive without it. So it's not necessary for life. And the last thing that you might see in some prokaryotic cells is a flagellum, and that's just something that's used to help them move through their environment. Now, on to eukaryotic cells. These are going to be a little bit more complex. They are highly compartmentalized, and I'm just going to quickly run through some of the organelles that you're going to find in here, and then we'll talk about them a little bit more in detail as we move on. So, starting over here at the top, this is a mitochondrion. That's the singular version. Mitochondria is the plural. Up here we have something called the cytoskeleton. Right here we have the nucleus. Here we have the rough endoplasmic reticulum. I'm just going to abbreviate for right now. Here we have our plasma membrane. And over here we have something called centrioles, which these are structures that were once thought to have a role in cell division, but we're now learning that they're not really necessary for that. Plant cells don't have them at all, and they're still able to divide perfectly well. And eukaryotic cells have been shown through different experiments to be able to divide without the centrioles there. So it's still kind of being studied exactly what those are for. Plant cells have a lot of the same organelles, but they do have some that are unique only to them. Up here we have ribosomes, which are found in pretty much all cells. This one up here shows the cell wall, which again is different from the cell wall we'd find in bacteria. This particular organelle is called a peroxisome. We'll talk a little bit more about that in a moment. Right down here we have our chloroplasts. Right here we have our Golgi apparatus, sometimes referred to as the Golgi body. We'll talk about what that one does. And then the last one I kind of skipped over here. This is the smooth endoplasmic reticulum. Very closely related to the rough, but it does have some different functionality. So all organelles are kind of divided into three big groups. The first group of organelles are found in the nucleus. And so this is where you're going to find the DNA and chromosomes and things like that. So if you look at this diagram, this is actually not a whole cell. This is a nucleus inside of a cell. And then inside of that nucleus is another organelle called the nucleolus. And this is where ribosomes are made. So they're going to be manufactured in the nucleolus, and then they're going to be shipped out to wherever they're needed in the cell. Everything else that's in the nucleus, all this granular looking material, that's our DNA. And we'll talk in great detail about DNA and what it does later on in the class. And then over here, these are chromosomes. So you can kind of see that standard X shape. Again, when we get to the unit on DNA, we'll talk more about chromosome structure and what exactly it is they do. But that's the nucleus, contains the DNA. It's the control center of the cell. Next, probably the most elaborate part of the eukaryotic cell is what we call the endomembrane system. And so endo just refers to inside the cell, and then membrane obviously is a network of intricate membranes. So this right here is the nucleus, and then right outside of the nucleus we have the endoplasmic reticulum. And from now on, we're just going to abbreviate that as ER. So at the endoplasmic reticulum, you have the rough ER, and what makes it rough is ribosomes. And those ribosomes are the site of protein synthesis. So the rough ER is involved in manufacturing of proteins, and then they actually get packaged into these little guys called vesicles to get shipped off to other places to do other jobs. On this side, we have what's called the smooth ER. So it's endoplasmic reticulum, but notice there's no ribosomes on it. The smooth ER is involved with lipid synthesis, and it also plays a huge role in detoxifying our body. Detoxifying. So as you can imagine, cells of your liver, which is the major filtration system of our body, or the detoxifying system of our body, cells in our liver are going to have a lot of smooth ER in them. The next part of the endomembrane system that I want you to pay attention to is this guy right here. And this is the Golgi apparatus. Not sure if I spelled that correctly, but the Golgi apparatus is kind of like the UPS department of the cell. This is where things are modified and packaged and sorted and prepared to ship out of the cell. So you'll notice kind of the flow of things here. We start in the nucleus. Proteins are made in the endoplasmic reticulum. They're packaged up, sent to the Golgi apparatus, where they get modified, sorted, prepared for shipping, and then they get sent out to the cell membrane, where they get expelled out into the rest of the body, wherever they might be needed. There are also special organelles involved specifically in energy production. Again, we're going to go into a lot of detail when we get to photosynthesis and cellular respiration, but these guys right here are mitochondria. So this is an actual electron micrograph picture of a mitochondria, and you'll notice all these little projections inside. That's what these folds are over here. And this just creates more surface area to carry out cellular respiration. Very similar to mitochondria are organelles that are found only in plants called chloroplasts. And this is the site of photosynthesis. The reason why green plants appear green is because they have these little stacks inside of them. These are called thylakoids. And that's where you find the pigment chlorophyll. And again, when we get to the unit on photosynthesis and cellular respiration, we'll talk a lot more about how that works and how these organelles control that. Those are the main organelles that you're going to find in eukaryotic cells. but there are some other ones that are worth mentioning. There's an organelle called a peroxisome. These are found in pretty much all eukaryotic organisms, single-celled or multicellular. To some extent, all cells contain these peroxisomes. And these break down hydrogen peroxide into water and O2. Obviously, I did not balance that equation, but generally speaking, peroxisomes break down hydrogen peroxide into water and oxygen. Hydrogen peroxide is actually a toxic byproduct of a lot of chemical reactions. So every cell produces it, and every cell also needs to get rid of it really quickly, which is what peroxisomes are there to do. Vacuoles are typically found in plants. They're going to be used for storage. So they're going to store water, nutrients, minerals, things like that. On the animal side, and we're talking more about single-celled things like paramecia and amoeba, you can have something called a contractile vacuole. And as the name implies, it contracts. And it can do that to regulate the amount of water inside of that single-celled organism, or it can actually be used kind of like an octopus or a squid, where it squeezes water and propels that organism through the watery environment in which they live. The cytoskeleton is extremely important for maintaining cell shape. It's made up of fibers called microfilaments and microtubules. So these not only give the cell their shape, but they provide kind of a highway, if you will, for cells to move materials around inside of them. So you'll see there's something called motor proteins that have this structure kind of like this, and it has two little legs, and these little legs will actually walk along a microtubule and they'll drag a big vesicle or something else with them, and they'll drag them around the cell wherever they need to go. And these little legs just literally walk along that cytoskeleton. And then finally, you might see some structures called cilia, which here's our cell. Cilia look kind of like little hair-like projections on them. Cilia can be used for cells to move. We have cilia on the cells that line our lungs to help bring mucus and dirt and things up from our lungs and get them out of our bodies. In single-celled organisms like paramecia, they can be used for movement, but they're just another organelle that you're going to see in a lot of different eukaryotic cells. So that's it for now, the difference between prokaryotic and eukaryotic cells, and we'll see you next time around.